Good evening,
Okay,
everyone. Yeah, so you can start. Yeah, please, Glenn.
okay, sir. Thank you, sir. Good afternoon and welcome to one and all. Myself, Rupsha from Clarinet, the designated session assistant for a seamless experience of the session. Clarinet is very proud to be a digital partner of this event organized by Indian Pharmacological Society, West Bengal branch. And the topic of today's session is online lecture session on basics of HPLC. Clarinet is India's most trusted and widely used digitech platform with multiple enriching services exclusively for doctors. So let's begin today's session, for which I would like to invite Dr. Samajdar sir. Over to you, sir. Kindly proceed with your talk.
Thank you very much. So, welcome all the respected delegates to our webinar on high performance liquid chromatography, which is a pivotal technique in the field of analytical chemistry. Today, we will discuss about this fascinating world of HPLC, exploring its fundamental principles and applications. And we are really fortunate that today we have with us Professor Samir Malhotra sir. Sir needs no introduction amongst the academicians, but it is a real privilege on behalf of Indian Pharmacological Society West Bengal branch to welcome Sir Professor Samir Malhotra, who had done his DM in clinical pharmacology. Currently, Sir is professor and head at the Department of Pharmacology at the esteemed institution that is PGIMER Chandigarh. So welcome, sir. And also, we are really uh, happy to welcome two of our faculties, Dr. Avaneesh Kumar Pandey, who is a PhD scientist D at a ICMR project at PGIMER Chandigarh. Along with also we are welcoming Dr. Ritika Kondel, a research associate of ICMR project from PGIMER Chandigarh. So welcome all. So let's embark on this exciting journey to uncover the secrets and efficiencies of HPLC. Welcome abroad. So I welcome Professor Samir Malhotra sir to take over the session.
Uh, yes, thank you, Dr. Shambo. We are glad to be here. Hope many people will join and listen to this. Our presentation will be sort of split into four parts. After a brief introduction, I will let the technical details be handled by the two scientists who are actually day-to-day working and involved in setting new methods and validation of methods. Then I will give talk about clinical applications briefly and we'll conclude. We hope to finish in about 45 to 50 minutes so that there are 5-10 minutes for questions if there are any. So I'll share the screen. Can you see the screen?
Yes, sir.
Okay, great.
Sir, just make it full screen.
At our side, it is full screen.
I think, sir, then one thing you can do, sir, you please stop screen share and again start sharing the screen. And at that time, you will get an option of that full screen. So you will click on that and we will get the view.
Now, what do you see?
Yes, sir. Now it's full screen, sir. Please, sir, please start.
Now, imagine there is a tunnel. Am I audible?
Yes, sir. You are audible, sir.
Actually, I have some throat issues, so I can't be very loud. I fell ill a few days ago and I have not recovered fully.
Okay, okay, sir. No, sir. We can hear you, sir. Take care, sir. Yeah.
Imagine you are standing outside a tunnel. There is a long queue, thousands of people. The tunnel is very interesting. it has alcohols of the best variety. Different varieties. You can go in, taste any alcohol you want. You can stay for as long as you want. If you are not interested, you can move out. As you can see at the top, some people moving out. So what will happen? There will be some people who will just walk and walk through the tunnel and go out. They are not interested. There will be some people who will be interested in having tasting a little bit here and there, like weekend drinkers kind of a thing. So they will stay for some time. They will make a few tastings and then they will move out. And finally, there will be some people who are, I won't say addicts, but really lovers of alcohol. They would not like to leave the tunnel. They will stick. So the people here are what we call the mobile phase. Alcohol is the stationary phase. So depending upon the affinity of the people for alcohol, we can separate the people among at least 2 or 3 categories. So you see, this is a very vast range of people to be separated, and it's very easy to understand that we can do that. But then, if you want to split further the big drinkers of alcohol, how to separate them? You need to set up some other experiment, and I leave that experiment for you to design. How will you separate those? And we'll move on to the topic. All of us has known or read about chlorophyll, the plant pigment. I have just taken the image from the Wikipedia page of chlorophyll. The leaf part, the green pigment that it contains. It was, more than 200 years, even more than that, people knew about chlorophyll and its importance. And people knew that there are different shades of green, and it's not one pigment, there are several pigments here. And if you read this Wikipedia page, you will see the names of several scientists who were involved in the research on chlorophyll. You will not see the name of one scientist, which I am going to tell you. Some of you might have heard about him, but most wouldn't, I guess. So this chlorophyll, people knew that it can be separated. For 100, 200 years, people were trying to separate chlorophyll into different components or different types of chlorophyll. They were setting up their experiments, elaborate experiments. Scientists, basically chemists, they were trying to do this, but all of them failed. Until this fellow came. He was a Russian, Mikhail Semyonovich Svet. He is known now as the father of chromatography. In fact, his surname Svet in Russian means color. So this fellow was, is responsible for discovering this. I will tell you a little bit of his life history. The story is the legend. I am not sure how true it is. You have heard of Einstein, looking at a falling apple and discovering gravity. The story here is that he was lying on his bed and he saw on the roof different patterns being made by the leaking water. And he saw that this is being separated, some rust or something else. There are some circles. He thought that I can do something with this. So he set up his experiment. He took chlorophyll and he dissolved it in a solvent, took a test tube, closed down one end or made it very narrow, filled the test tube with chalk and poured the mixture of alcohol and chlorophyll on top. And after that, he added more alcohol. In the alcohol experiment in the first slide that I showed, if there are some people who are not willing to move out, they need to be pushed. Some pressure has to be applied. So in this case, he applied pressure, very low amount of pressure by just putting some more solution without the solute on that. And there was a brilliant separation of a few, compounds, and he could very clearly see that chlorophyll is different. So this was his original experiment. This is from his paper that he published uh, more than 100 years ago. There was his column. He in fact went on to design a very elegant apparatus in which in one set he could use several columns, a small column, larger column, columns of different widths and things like that. So that he could test various parameters with which you can separate chlorophyll and how to do it best. And he did separate. He published extensively, but unfortunately those publications were either in Russian or German journals, and English speaking literature, somehow people did not know much about him. In fact, when he presented his findings at the Russian conference, people did not understand what he was talking about, and the famous scientists of that time, they did not believe what he was saying. They said it's not correct. It cannot happen because they had spent decades of their lives trying to separate chlorophyll using many experiments. None of that succeeded. And using simple chalk, it is believed that chalk, because you color the roof with chalk. That's why he took chalk. Uh, and with that simple apparatus, he could separate chlorophyll into different components. But his life was a tragedy in the sense his mother died a few days after he was born. His father was traveling all over Europe. He studied in Switzerland, where he did get a degree. But then, when he came back to Russia, at the time Russia was ruled by an emperor. So the Russia did not recognize that degree, and he had to start from as a lab assistant. It was as lab assistant that he discovered chromatography, and he gave this name chromatography. Uh, he knew a little bit of those languages, Greek or Latin. Otherwise, he could have called it colorography, but he called it chromatography. Uh, he he married. It said that his he was not very healthy fellow. His wife had to take care of him a lot so that he could work. But misfortune kept always targeting him. When he thought that he is settling in Russia in the lab of a very famous scientist, from lab assistant, he managed to get a degree there as well. But what happened was, those days there were many protests against the emperor and the czar, as they were called. And once there was a protest by the students in the university. Now the scientist who was his mentor, or in his lab, in whose lab he was working, that professor went on to support the students. He collected some signatures of other faculty in support of the students. Naturally, the authorities did not like it, and his lab was closed. That professor was thrown out of the university. So after his lab got closed, he had to start all over again. He had to go to Poland, where he started again. He started to repeat his experiments, refine his experiments. But then First World War started, and he had to evacuate from there also. Unfortunately, he had all his data and records were there in the university, but he was not there at the time of evacuation. So the university managed to save a lot of their papers and documents, but nobody gave importance to his papers. So everything went into the hand of the enemy, that was Germany at the time. He lost everything again. Uh, then he went back to Russia, and after that his health started to deteriorate, and he just passed away a few years later. And the slave has a very, the grave has a very interesting inscription. He invented chromatography, which separates molecules but unites people. Later on, as I said, Germany was a very important figure in this field of chromatography. I am not sure whether any documents that they had seized from the Polish lab of this scientist, were they of any help or not to Germany in setting up their own labs and in this field. He was nominated for Nobel Prize, but the Academy did not award him. It went to some other scientist at that time because chemists were not very happy with him because he was a botanist. So chemists always said that how can a non-chemist come up with some system or some method that can that can be of help. And after his death, he was virtually forgotten, even in his own country. It was in the later decade when Nobel Prizes started to be given for discoveries of isolation and preparation of vitamins, hormones. All of them had used chromatographic method. Then people started to wonder who was the person who started this. Then everyone came back to him. Unfortunately, he was no longer there to answer any of the questions. Now we come to the technical part of this, for which I will request Dr. Avaneesh to start and take over.
Thank you, sir. Sir gave us a great foundation for this talk. So now I will talk about the technical part of chromatography. So what do you understand by chromatography? As sir said in briefly about the chromatography, but in technical terms, chromatography is a analytical science of separation of one molecule from the another in a mixture at the molecular level, which in a liquid solution or in a gaseous state. So what is the fundamental principle underlying the chromatography? The fundamental principle of chromatography is the separation of molecules on the basis of interaction of liquid, that mobile phase as well as the stationary phase. So on the basis of that, their principle which is involved in the separation of molecule, it is categorized in two parts. One is adsorption chromatography and another one is partition chromatography. In adsorption chromatography, the stationary phase always a solid phase and mobile phase is liquid. So the analytes were separated on the basis of their different degree of affinity to adsorb on the stationary phase, and so that affinity of adsorption on the stationary phase leads to the difference in the rate of movement in the molecules throughout the column, and further it leads to the separation. Like in the example, there is a two compound X and Y in the solution. X has more affinity with the stationary phase and less affinity with the mobile phase. So it retained for more time in the column, and on the other hand, Y has the more affinity with mobile phase and less affinity with stationary phase. So it is moved faster and it comes out first from the column. On the other hand, in partition chromatography, the stationary phase is liquid coated on the solid support, and mobile phase also a liquid. So here the separation will happen on the basis of distribution of molecules between stationary phase as well and mobile phase. The different degree of distribution of different degree of solubility of compounds in both mobile phase as well as in stationary phase makes the different rate of movement throughout the column, and further it leads to the separation. As here in the example, there are a solution of amino acids added in the column. Column is the packed silica coated with water, and mobile phase is chloroform. So all the amino acids were separated as per their distribution between water and chloroform. So this is the basic principle for the chromatography. So there are different type of chromatographies, like gas chromatography, in which the mobile phase is gases, and it is used for the identification of volatile compounds and gaseous compounds. The next one is liquid chromatography, which is the most used form of chromatography, and in which the stationary phase is fixed and solid phase, and the mobile phase is liquid. So next one is TLC. It is thin layer chromatography. It is a form of liquid chromatography in which the thin layer of stationary phase are coated on the solid support, and once the liquid mobile phase moves through against the gravity through the capillary mechanism, it will create the separation of molecules from the mixture. Next one is from paper chromatography. It is also a form of liquid chromatography in which paper acts as a stationary phase and the solvent acts as a mobile phase. Flash chromatography is a different kind of chromatography which is used for the purification of compounds, in which the column is put on the more than 200 psi air pressure that makes the faster purification of the compound. And further, high performance liquid chromatography is a form of liquid chromatography, which is further I will discuss in detail. Next one is ion exchange chromatography. It is used for the separation of ionic compounds. And next one is UPLC. It's a ultra performance liquid chromatography, and it is also a advanced form of liquid chromatography. And there is a next one is RPLC. It is a reverse phase liquid chromatography in which the chemistry of column is different from the normal phase liquid chromatography, which I will explain in coming slides. So initially, HPLC always people know it as a high pressure liquid chromatography, because traditionally the liquid chromatography the mobile phase was pushed at very low pressure. So it takes much time to separate the molecules in column. Once HPLC came in the picture, there is only one additional advancement is the high pressure. So initially people started naming it as a high pressure liquid chromatography, because high pressure creates the faster and efficient separation of molecules in the column. But after some time, due course of time, the advancement in instruments and the advancement in special advancement in the chemistry of column created increased its capacity, increased its capacity. So people started calling it high performance liquid chromatography. So I will discuss some terms which is used for further in the talk, and it is used generally used during the HPLC analysis. So sample. What is the sample? Sample means any mixture or any compound which is injected in the HPLC for the identification and quantification of our compound in which we are interested. So it's a mixture, or it may be a alone compound. It may be biological sample. It may be pharmaceutical compound or pharmaceutical formulation. It may be anything. So next is analyte. Analyte is the compound to which we want to identify and quantify by using HPLC. Mobile phase is in case of liquid chromatography, mobile phase is always a liquid, and it is the carrier for the sample which carries the sample throughout the HPLC system, and it provides the opportunity to analyte to interact with the stationary phase, that is in some cases it is mostly silica, and as interact with stationary phase and got separated during the movement. Stationary phase in case of liquid chromatography, it is a solid phase, and it is packed in a column, and it will provide the surface for the interaction of analyte for the separation. Resolution is it's a degree of separation between two adjacent peak. More the difference in two adjacent peak called more resolution, and less difference in time is called less resolution. Retention time is the time taken by any analyte for in that getting eluted from the column is called as retention time. It is usually called it is the time between the injection and the detection in the detector. So it is called as retention time. Internal standard. So internal standards are structural analog of the or interstate compound of interest. So it is added in a constant amount in each sample, and it is added because to ensure it is added for the ensuring the consistency of the method in detection of consistency in the performance of the method. So coming to the HPLC components, there are multiple components. First is the solvent reservoirs. In advanced machines, nowadays companies provide two or four solvent reservoirs. So we can use multiple mobile phase for the analysis. Next is the major part is the component. That pump comes with the solvent manager. It means gradient manager. You can say pump is responsible for the taking mobile phase from reservoir and pumping it throughout the HPLC at constant rate and a constant pressure. Solvent manager manages the proportion of the component. Proportion of the component of the mobile phase. If there is two part of the mobile phase, like one water and one alcohol. So solvent manager is responsible for the maintaining the proportion of solvents as recommend already as per the command. Next part of HPLC is the injector. Injector is responsible for through injector we inject our sample on the column, and it may be manual, it may be automatic. Once it is automatic, we call it as a auto sampler, and in case of manual, for every sample we need to inject one by one, and in case of auto sampler we just put all the samples in a tray and just give the command that machine will take automatically one by one and inject it to the column. Next part of HPLC is the stationary HPLC column. It is a packed material and it provides the stationary, it works as a stationary phase for the HPLC. And it is that column is mainly responsible for the separation of molecules in the sample. So after HPLC column, there is a detector, which is it is responsible for identifying the compound eluted from the HPLC column, and it on the basis of its functioning, it creates a electronic signal and which is recorded in the computer data center in the form of chromatogram. So these are the basic components of the HPLC. So on the basis of mobile composition of mobile phase, that HPLC operations are divided into described as isocratic as well as gradient. So isocratic means iso means same. It means the composition of the mobile phase stays same throughout the process of the analysis. It may be a single compound, it may be single solvent, maybe water, maybe alcohol, or maybe the composition of 2 or 3 solvents in a fixed ratio. So this operation is called as isocratic. Gradient means the solvent compositions change throughout the run. It means initial 5 minutes maybe 20 as to 80, and next 5 minutes it will maybe 40 and 60, and next is 90 and 10, and then again it will reverse. So it will change throughout the column, throughout the process, throughout the run, and it pattern of change or change in composition may be stepwise or linear gradient, yeah, gradual change. Stepwise means it will change, sudden change in the composition, and gradual means it is changing gradually, little by little. So on the basis of column chemistry, chromatography is also defined. Chromatographic operations defined in two forms: normal phase chromatography and reverse phase chromatography. In normal phase chromatography, stationary phase is polar in nature, more polar in nature than the mobile phase. And this form of chromatography, that normal phase chromatography is generally used for the identification or separation of polar molecules like vitamins, steroids, and other. And in case of reverse phase chromatography, the stationary phase is less polar than mobile phase, and it is mostly used. It is the most used form of the chromatography, and it is used for non-polar molecule. But the reverse phase was normal phase is converted to reverse phase by using the non-polar carbon chains. Here C18, we always say C18 column or C8 column. C8 or C18 means the silica molecules are capped with the non-polar C8 carbon chain or 18 carbon chain. So that carbon chains make it less polar, and so it is called as reverse phase chromatography. So now we will understand with the example how the interaction of stationary phase and mobile phase makes the separation. In this case, there are three compounds: yellow, red, and blue. So yellow one has more affinity with the mobile phase and less affinity with the stationary phase. So it is moving fast, and it will elute first from the column. On the other hand, the blue compound has the more affinity to attach to bind with the stationary phase and less affinity with mobile phase. So it will move slow, and it will take more mobile phase to get elute out from the column. So here we can say, once as yellow compound is moving fast, so it will elute first from the column, and because it has more affinity with the mobile phase, so it will take less amount of mobile phase to elute out, and so it will show the sharper peak. And on the other hand, once the blue band has more affinity with the column, and so it takes more mobile phase to get elute out, and so it will show the broader peak. So there are two basic physical parameters of the column which affects the resolution of peak. So first is length of the column, and second one is the particle size of the packing material. So smaller in the length of the smaller length of the column gives the lesser time to analyte get interacted with the stationary phase. So if it will interact for lesser time, it means it will not separate properly. So if you increase the column length, it means you are providing the more time to the we are providing more time to analyte get interact with the stationary phase, and so it will get separated properly. On the other hand, if the particle size of the packing material is bigger, so it means if particle size is bigger, it means surface area of packing material exposed to the analyte is less. So in that case, the analyte has less surface area to get interact. But on the other hand, if we decrease the particle size of the packing material, it means we are increasing the surface area, exposed surface area to get for the analyte to get interact. So it will cause more interaction. So more interaction leads to better resolution. So coming to the detector part, it is the last and part of the HPLC. Here, in case of UV detector, we specify the specific wavelength to get pass through the lens on flow cell. So that and specific compound have the nature to absorb a specific wavelength, light of a specific wavelength. So once the light put on flow cell, that molecule absorbs some amount of light, and remaining will pass through the flow cell and get on the photodiode. That photodiode will convert this signal in an electronic signal, and that in the electronic signal will converted into the chromatogram in the recording in the computer. So there are other detectors used for the used in HPLC, like PDA, photodiode detector. It is also called as a diode array detector, and another one is fluorescent detector, and another one is that mass spectroscopy. It depends. Detectors are use of detectors depend on the nature of the analyte. So till here we have discussed about the principle and the basic components of the HPLC. Now we will discuss if we want to develop HPLC method for the analysis. So before starting our method development, we need to have some preliminary preparation. For that, we need to do the thorough literature search for checking the property of drug and its PK profile and its physiochemical properties. We should purchase reference standard and with its certificate of analysis. We should prepare some stock solution, and we will divide the stock solution suitable in liquids, and we need to check for the instrument availability for next 7 to 10 days, and we also check the column life for 100 injections, and also need to check the availability for solvents and reagents. So once we have identified our HPLC method for further analysis, before going for further analysis, we need to check the functioning of our HPLC method. For that, we need to generate the calibration curve. For generation of calibration curve, we need to run at least 8 samples, in which 6 samples of known concentration, which covers the range of quantization range of which we need for further analysis. One blank without IS is internal standard, and one with the internal standard. After running these samples, we get the AUC from AUC, and we will plot this AUC against the concentration, and we will generate a simplest regression model. Once the regression, that linear regression model was developed, and we check for the R square. If R square is more than or equal to 0.99, we can say our method is okay, and we can go for further validation and testing. So during the development of method, we face some troubles, like some problems with baseline, like we see the noisy baseline, cyclic baseline, spikes in baseline, drift in baseline. So noisy baseline is basically due to the air bubbles present in maybe in mobile phase, maybe in air bubbles present in pump or any tubing, and noise also can come due to contamination, due to bad condition of column, or bad preparation of our sample. Cyclic baseline comes when there is a fluctuation in temperature of column 1. Spikes we get once the air bubbles are trapped in the head of detector. Drift, we see drift in a baseline usually during our gradient method. Once the one part of the gradient mobile phase has more affinity to absorb UV light, so it will once the concentration of second part increase, that baseline goes up and it causes the drift in baseline. There are the other problems like peak fronting. Peak fronting means first part of the peak is more broader than the last second part of the peak, like here. So it happens because once we use mobile phase which is not suitable for our in which the analyte is not properly have properly dissolved in the mobile phase. So for resolving this issue, we can decrease the amount of injection volume, or we can decrease the concentration of our sample. Tailing peak, peak split and soldering and peak shift, these problems are due to the bad column condition, bad sample preparation, more viscous samples, contamination in samples, bad condition of column, or less washed column. And another problem is, and for these we can check for the column life and proper washing and better purging to the system. And last problem we sometimes we face fluctuation in pressure. So in that case, pressure fluctuation is directly related to the performance of pump or condition of choking or condition of the column. So these are the basic troubles, and these can be sorted out. Thank you.
So, yeah. So after having an explicit understanding of the basics of HPLC and the method standardization we need, so let's go further. So we'll now understand what is the bioanalytical method validation. So let's understand what is bio means here. Why we call it bioanalytical. Bio means that the method, so the metric system where you need to identify or analyze the molecule of interest. It is of the biological origin. So it could be blood, it could be urine, it could be CSF, any component of biological origin. So, and what validation means here? It means that the process in which where we see the reliability of an analytical method to quantify that analyte. So its aim and objective is that it is a documented evidence that suggests that the method we follow, it's a fit for intended purpose, and the results are of quality, reliability, and consistency. So let's understand this in a simple language. Like, for example, we have a recipe for a dish and we want that dish to be made by us. So what we need, what we need, that we should replicate that dish and it should be of same taste, same quality. So which will be ensured by this validation process by following some steps. So there are some guidelines for a bioanalytical method validation by the FDA and ICH. And my further slides will be from these guidelines only. And so they have illustrated the various steps to be followed while validating a bioanalytical method. So these are some regulatory requirements, like before having any method, any bioanalytical method to be standardized or validated, one should follow a good laboratory practice in their lab. So which will include the preparation of SOPs, starting from the sample collection to reporting. So which will include the record keeping of the samples to be kept in a refrigerator, which will include the sample preparation protocols, the analytical method, like whether you have used the HPLC or you have used the LCMS, then the procedure followed for the quality controls and the record keeping. So all of SOPs and GLP practice should be followed while developing a method for bioanalytical sample. So let's understand the workflow. Like as for let's assume that we want to analyze our drug in a blood. So it could be analyzed through blood or through the plasma. So first we will do the extraction procedure. We will precipitate the protein, and we will develop the method. So this method has been taken by the Dr. Avaneesh, and he has explained what needs to be considered while developing a method of HPLC. Then comes the process of validation. So these all are the parameters which need to be undertaken in steps, and we will go in detail of these steps in further slides. And after having this validation, we can now further proceed to conducting any study like a bioequivalent study, then pharmacokinetic, or a simple pharmacokinetic study of a particular drug. So there are types of validation. Full validation, when you have a new molecule or a new drug to be analyzed for the first time, then full validation procedure or steps need to be undertaken. But and there are the second type is the partial validation. If some minor changes are to be made in a pre-validated method, like for example, you have changed a protein precipitation procedure for a drug, or you have changed any detector system for a drug, then we can have a partial validation in that case. Then cross validation, if like for example, if your samples to be analyzed in two biological labs, one sample, in that case we need comparison of the validation parameters in between two labs. So there are some steps, there are some parameters which need to be assessed before conducting a inter-lab detection. So these all are the validation parameters which should be considered while developing a bioanalytical method validation. So first is the linearity or calibration curve. So it just means that we should follow a simple regression model where the concentration it's to be linearly plotted with the area response, and the R square should be 0.99. And while conducting any of these further parameters, this calibration curve it should be run. Then second is the accuracy, precision, recovery or matrix effect, then selectivity, sensitivity, and stability. So we'll understand each of these parameters. But before that, we need to understand some of the terminology. The terminologies which are used while discussing these parameters. So this is a limit of detection, LOD. What do you understand by this LOD? This is the lowest limit of a concentration of a drug which is detected by a system. System means any HPLC or a LCMS. So here there's a difference of detection and quantification. So detection means that you just see the peak of a drug. You are not able to quantify that. Then this LLOQ. So this is a lower limit of quantification. It means the lower concentration of a drug, and which could be quantified by an system, by an HPLC. So here I want to clear one thing, that all these are the concentrations among your calibration curve. So they are derived from your calibration curve, which was standardized during a method development. And upper limit means the highest amount of concentration detected by a system. Then we have some quality controls, which is LQC, MQC, and HQC. This LQC, it should be 3 times the LLOQ. Medium means that the medium value of your calibration curve, and the HQC means it is the high. We should not call it higher, but a concentration less than the upper limit of quantification. So these quality controls we have to decide among our calibration curves, and the range will depend on the concentration which you want to have in your in your study. So most of you have all have seen this accuracy and precision dart and board experiment. So we'll understand this by this dart and board experiment. So here we have 4 cases. What is accuracy means? It is the closeness of your closeness of it of a determinant value to a theoretical value. So we see in this third case, this is highly the our hits are highly have high accuracy to a bullseye, but it has low precision. Why low precision? Because the closeness of repeatability is very far. So here, in that case, this has high precision. And then in this first case, it has low accuracy and low precision. And this fourth one, we all know that it has a high accuracy and high precision. So, what is the acceptance criteria? How to decide what is how much is accuracy, how much is accuracy permitted, and how much precision permitted? So we have to run different these quality controls samples for 3 consecutive days and in different independent runs to see. So for accuracy and precision, we have to run the 5 independent run of this LLOQC, this quality, all 3 quality controls, and then we have to calculate the mean value of all these independent run, and it should be within the plus minus 15% of the theoretical value, except for the LLOQ, which should not deviate between more than 20%. So, for example, we can see that this is an accuracy calculation we have performed for one of the method here. So these are the independent runs. This is for intraday and this is for intraday. And we have calculated the mean, and then we have seen the error. So it is less than the 15%. Or less than the 20%, sorry, for this, because this is an LLOQC. Then how this precision, what is the acceptance criteria for precision? It is, we know that it tells us about the closeness of the replicates of run. So here we check for the coefficient of variation, and it should be less than 15%, and for LLOQ it should be less than 20%. So what precision will tell us? It will tell us about the repeatability, reproducibility, and the ruggedness of our method, like how accurate our multiple runs are. Then, so this is our precision calculation. So here we will just check for the coefficient variation for both for the intraday and the interday. Now coming on to the next third part of the full validation parameter, that is the recovery. So what recovery will tell us? It will tell us about the extraction efficiency of our analytical method. So what will we compare here? We will compare here the procedure followed for extractions. Like for example, you have diluted your sample with acetonitrite. So here we will compare the results with the post-extracted plasma, where the extraction procedure has been undertaken previously, spiking the drug, and in this case, after spiking the drug. So it will tell us about how efficient our extraction procedure is. So here, this is post-extracted and extracted. So we will compare, we will divide that area, and we will check for the recovery. So the recovery, it should need not to be 100%. However, it should be consistent and reproducible. Then coming on to the next part, that is the matrix effect. Here we want to see that how efficient our method or developed is efficient in quantifying the drug in another matrix. So for example, we will calculate in a aqueous form, we will divide the area in aqueous and the post-extracted plasma. So here we want to see the change or effect of a bioanalytical method in the presence of a matrix, that is the plasma, for example here. So we will compare the area and we'll see the change. We'll see the percentage. Here, we'll see the matrix effect. We'll divide the area and this matrix effect percentage. So now coming on to the last 2 parts, that is the selectivity and sensitivity. So selectivity here means that how efficient is a method. It has the ability to analyze the molecule of interest in presence of other compounds. Like for example, let's take an example where, for example, you have gone to a library and the librarian tells you about the book position. So how efficient was librarian to tell you that where the book is kept in the bookshelf? So that will tell us about the selectivity. So we will take different lots of plasma from different persons, and we will inject the LLOQ sample in it, and we will see that we will see the variation between all these, and it should be less than 20%. So it will tell us about the selectivity of your method. And for the sensitivity, it was analyzed by our lower limit of quantification. So how sensitive is your method to detect the lowest concentration? So this is the sensitivity. So, and I have not discussed about the stability parameter here because it will be a lengthy procedure, but it has the same values only. The accuracy was determined in many conditions like the freeze thaw condition, the auto sampler condition, but the values or the acceptance criteria remains the same. So that's all from our side. So, sir, yeah.
So very briefly, I think, of course, it appears complicated and complex, all these procedures, but once you start doing them, you know, become used to them and things start to happen in a better way, and you will start to learn as well as do the things. You will find that, you know, one day you have standardized the method, you have validated, done everything. Next month you are doing it, results don't come. So every day there are new challenges in this. Regarding application, you know, there is no field in the world which does not use the applications of chromatography, like analyzing samples from ocean or deep inside the earth or in the space or in nuclear physics, like, you know, nuclear bombs. Chromatography is everywhere. In our field of medicine, you know, forensics is one important area. Many of the methods of estimation of levels of proteins and several compounds are HPLC based or chromatography based at least. Finally, in pharmacology, drug development, you know, whenever a new chemical entity comes, its methods need to be developed and validated. Then in TDM, therapeutic drug monitoring, of course, it's a day-to-day application for us. You get some patient samples, they need to be analyzed. Of course, each of these is, you know, in itself a separate session. For example, TDM or drug development. I'll just conclude by saying that, you know, many times people will tell you in institutes that these are expensive equipment and their running cost is high. But you have to understand that even though it's true, some of the techniques used in, for example, immunopathology or even microbiology, they are no less expensive. There they are routinely being used. For example, in the case of tacrolimus, you know, it needs to be done. If your institute has transplantation occurring, tacrolimus levels will need to be estimated. And your departments can take a lead in that, and I think they should. You have to fight for, you know, the cost kind of thing that, okay, this is expensive, but it justifies the cost. With these words, I would like to end the session and thank you all for listening.
Thank you, sir. Thank you very much. Thank you also to Dr. Avaneesh ji for your wonderful deliberation and Dr. Ritika ji. So thank you on behalf of IPS West Bengal. So may I request now Team Clarnet, if you have any questions in the chat box, can you please share with that?
No, sir, we haven't received yet anything.
So I'm requesting all the delegates. I think we have almost 160 delegates who are viewing this live. So may I request all the delegates, if you have any question or queries, you please drop in the chat box so that that can be addressed by our respected faculties. So, sir, we will wait for maybe 1 or 2 minutes like that. If there is any question is coming.
I'll be here. I have another meeting, but I'll be here. I'll be here in the room only.
Okay, sir. So, thank you. One thing just want to ask to the respected faculties, as Malhotra sir is already told us about the application in TDM. So, what are the basic prerequisite we should have in our departments who want to provide this as patient care service? And if you have any idea on what are the important drugs we should choose fast as a patient care service uh, when we are providing TDM.
Uh, so like for running a TDM, like for in our department also, so the basic requirement is having an HPLC machine. So it could be auto sampler, it could be manual sampler machine. So it's the simplest conventional HPLC would work, and you just need the standards of the drug which needs to be analyzed, like for drug like anti-epileptics, like for example tacrolimus, any anti-infectives, like for example voriconazole, where the concentration needs to be taken, consider vancomycin. So you just need standards of that drug, and one personal like lab technician or one scientist who can prepare samples and who can precipitate the do the extraction procedure. These are the prerequisite for having TDM to be run in a department. Yeah.
And how many samples per day for if one machine we are using? So how many samples we can have? And that means the volume of the patient care facilities, if you can let us know about some idea on that in each machine.
Yeah.
We need 2 ml 2 ml blood sample is more than enough for the analysis for the drug.
I am asking about per day, how many patients we can serve like that?
Yeah. So it will depend on the extraction procedure of a drug. Like for example, for a drug which need more further steps, like it takes 2 hours for a method to be extracted. So in that case, we can analyze like 10 samples per day, or for a drug which needs just a simple procedure, so where 20 patients can be
And
analyzed.
it also depends on how many drugs we are trying to analyze. Like if we are just analyzing tacrolimus, you can go for 20 or 25 samples also. But if we are analyzing tacrolimus in 10 samples, and you are doing phenytoin in 10 other samples, so if you have a single machine, it won't be possible to do on the same day. So you need to do it in consecutive alternate day, or you can day.
If there are same drug samples, several samples, and you have an auto analyzer, then it's easier.
Median.
If then you can have auto analyzer, then it's difficult. You have to manually inject.
Yeah. If you have it on auto analyzer, you can go for 50 or more than 50 samples for one drug. If you are analyzing different drug in different samples, then it will take, because machine
column
washing, preparation.
Column change.
Yeah. Machine preparation takes 2 to 3 hours, at more than 2-3 hours. So it is not possible on same machine on the same day. But
sometimes, there are research samples for the projects that people are doing. They have also to be accommodated. Then there are all different drugs. Then patient samples are there. So we have separate days for this. Faculty member gets different days, each faculty member. Then there are days for patient TDM, then research samples.
Okay, sir. Thank you, sir. I think one question is like that from one of our PG student. He's asking that if you can little bit explain that chromatogram the x-axis versus y-axis. If you just explain that, if it is possible.
Yeah, in
Calibration curve.
are you
I think he means calibration curve.
Yeah. No, he is specifically asking about the like HPLC analysis is a chromatogram. So this graphical representation where x-axis and y-axis, x-axis measures the time and y-axis indicates the intensity. So how we can explain in different analytes. So like this he had mentioned.
Yeah. So a chromatogram is the it's a graphical presentation of the uh, detect detection of the analyte. Once the analyte comes out from the column, it is detected by uh, the detector. So it starts with zero and it goes up to peak and then comes down. So this procedure called the peak and area under the curve is analyzed, and area under curve represents the amount of drug. So that area under curve will plot we plot area under curve against the against our put in our algorithm developed by our calibration curve, and then we can convert this AUC in the actual concentration.
Which AUC calculation is done by the machine.
Yeah.
You don't have to do this.
Okay.
Just we need to put that AUC in our calibration curve, calibration equation. Yeah.
Okay, sir. So another question asked by, I think, Malhotra sir can address this. Dr. Gramle Amol, he is asking, is HPLC training that is included in the postgraduate pharmacology curriculum? And what examiners are expecting about the...
You see, I am not aware of NMC curriculum, whether they have it. We don't follow NMC, we don't need to. But our residents do get training in HPLC, mostly as part of TDM or some of the research projects. They do not sort of get to manually use the equipment, but they have to be there. They have to process the samples. So there is an expert technician or these scientists who actually handle the equipment, but they are expected to know the basics, the method development and method validation. These they are expected to know.
Yes, sir. Okay. Okay, sir. So I think we don't have any more question. So once again, we are really thankful to the team of PGIMER. Especially, we are really thankful to Professor Samir Malhotra sir for agreeing and guiding this whole HPLC program. Yes, it is important to have some video demonstration of HPLC works. We will definitely keep it in our next program so that we can have some video recorded procedures. So thank you again, sir.
During the annual workshop that we conduct, we demonstrate these things to the delegates. So they do get some exposure. I think people came from your state as well this year as well. We also have an LCMS which we did not sort of discuss today because there wasn't enough time. But that's again a totally different topic. Thank you for giving us this opportunity. I'm sorry I have another meeting waiting for me. So we'll leave. Thanks.
Oh, another question asked, I think, can we take that question last? Dr. Priyanka Bishwas from West Bengal. She's asking for detection of mycotoxin, pesticide, or antibiotics in feed or tissue sample. Do we need to use separate column?
See, first of all, like pesticides, they are estimated using gas chromatography. HPLC doesn't work on them. So mycotoxins, I don't know how they are detected, which equipment. Antibiotics, you can use HPLC to detect many antibiotics. Regarding what was the other point? About tissues. See, you have to standardize the method of extraction from the sample, whatever the biological tissue. In theory, it can be done from any tissue.
Okay, sir. So, thank you once again, Professor Samir Malhotra sir, Dr. Avaneesh Kumar Pandey sir, and Dr. Ritika Kondel madam. So, for giving us this wonderful opportunity to host you all. And I think all our postgraduate students, they will be immensely helped by this introductory lecture. And I think many difficult concepts had been discussed very well in this lecture series. So thank you once again, and we are really hopeful that in the maybe some next meeting, we can discuss some other important topics regarding this postgraduate pharmacology training under the guidance of you, sir. So thank you once again.
Thank you so much, everyone, for extending us the opportunity to host a session. So with all your permission, I would like to sign off here. Thank you.